All right. Well, I think we've, given them a little more time to log in and get all of their audio set up. So I'll go ahead and get started. Howdy, everyone. My name's Abigail King. I am lucky to be this seminar's postgraduate student, and I got to invite the lovely Dr. Nichola Hall. Um, First off, I just wanted to talk about kind of what our seminar is about. So horticulture sciences is becoming a hotbed for application of technological innovations and a place for new innovations. From the use of drones in the orchards and vineyards, new approaches to harvest, and the development of new genetics, innovation is needing to meet the needs of sustainable production of foods. This is more important in the ever-changing economic and weather patterns. Therefore, the theme of this semester's seminar is Innovations in Horticulture. I'm excited to introduce to you our first speaker of the semester, Dr. Nichola Hall. Dr. Hall is originally from Scotland, where she obtained her undergraduate degree in microbial biotechnology and her PhD in yeast physiology. In her over 20 years in the wine industry, she has spent time in wine production, wine quality, as well as enological supplies. Dr. Hall is very involved in the winemaking industry and is involved in wine industry groups. She is a past president of the American Society of Enology and Viticulture, known as ASEV, and past chair of the United Wine and Grape Symposium. I had the pleasure of meeting Dr. Hall this summer at the most recent ASEV meeting in San Diego at a mentoring lunch. I was so fascinated with her wine interests and industry work, I knew we had to have her for the seminar series. Dr. Hall's winemaking interests vary, but she has a passion for aromatic whites, and in her free time, she spends it golfing, skiing, and enjoying the California sunshine. Without any further ado, Dr. Nichola Hall.
Thank you, Abby, so very much for the lovely introduction. So as they say, right, no good deed goes unpunished. So we met at the mentorship lunch, and here we are. So it goes to show the value of our industry, our conferences, and our networking. Because without that meeting, right, we wouldn't be here broadening our horizon. So it's absolutely my pleasure to be here. I would love to have been there in person, but just with the harvest schedule, my schedule was a little tight. But as I've said, my name is Nichola Hall. If I say anything today and it's the accent, let me know and I can repeat it. If I say anything today, it's not the accent, but the terminology, or it's just something that you're like, wait, what did she say? Just ask me to repeat it. But we're going to chat a little bit today about the role of non-conventional and conventional yeast in winemaking. So you're all horticultural students, right? So not many people have their camera on, but if you could kind of show and whatever type of reaction you want, how many people have been involved in winemaking? We got one, two, three. You're all moving about the screen. So we've got quite a few, right? Winemaking is changing. When I started in the industry, the hardest thing was to get bacteria to grow in the wine. Our wine chemistry was so different. Here, 2 decades later, we're like, what are all these bacteria and what are they doing? Right? Organisms will adapt to their environment. And their environment is really, really changing. So just before this, I was just in a meeting with my ACB colleagues planning a climate change symposium focused on enology. And we're talking about the grape matrix is changing. The plant matrix is changing. Our chemistry are changing. How do we adapt our processes to accommodate these changes? How do we have the same outcome? How do we get to the same place with all our matrix being different? And a big part of that is our microbiology. We could split winemaking up into microbiology, chemistry, and sensory at the very basics. So what we're going to do today is talk about the innovations in microbiology in order to help you kind of optimize wine quality. So those of you who are actively making wine, interested in making wine, I hope this is of interest to you. Those of you who it's maybe a little bit new to you, maybe will stem a new passion for you. So I'm going to share my screen. And as I said, if there's anything you don't understand, just kind of jump in. Oh, Abby, you have disabled screen
Oh,
sharing, or the host.
let me,
We didn't do a little check, did we?
I know, we just were chatting.
We just chatted. So yeah, so that's what we're going to talk about, right? These innovations. So initially, I'll introduce to you the brief introduction of aromatic compounds in wine grapes, because we're talking about wine quality and preserving wine quality, optimizing wine quality. We're really talking about its aromatic qualities. Talk to you a little bit about the yeasts that are involved in the winemaking process, both those traditional or conventional yeasts as well as some non-conventional yeasts, and how that we are really using them to enhance our wine quality. We'll talk a little bit about Saccharomyces. We can't forget our mainstay organism, and then we'll have a Q&A session. Saccharomyces, Salmonella, Parlospora, blah, blah. And with the asexual forms, they're generally candidas. And we acknowledge that these organisms that come around sound grapes have got a role in the pre-fermentative stage. When we say pre-fermentative stages, winemakers, we're talking about the stage from harvest up to about 2 to 3% sugar depletion. And we acknowledge that these organisms can add complexity to the final wine aromas. We'll say, well, they can impart really desirable fruity aromas. They can give you nice floral attributes and lead to some really nice textures. And that's when you've got the correct assortment of non-Saccharomyces yeast there. So non-Saccharomyces is a colloquial term for these different yeasts from these different genres. And when they're working together, they have been described as like an orchestra. They're all playing a role, and their individual role is important, but how they are working as part of a group is even more so. But sometimes they don't work all together and produce these beautiful, desirable fruity and floral aromas. Sometimes they produce a lot of really undesirable aromas and textures. Sometimes these organisms, under certain circumstances, like if the fruit is too hot, if the pH is too high, if the skin of the grapes has been damaged in any way, they can take those wonderful grape sugars we've worked so hard to get, and they'll put them through a series of biochemical reactions, and we end up with things like vinegar. We'll end up with things like nail polish removal. So we'll have acetic acid, ethyl acetate. We can have a lot of oxidation occurring, very high acetaldehyde levels. So our beautiful grape juice tastes like bruised apples. And they could produce a lot of other compounds that lead to unpleasant mouthfeel, unpleasant textures. And the challenge is we don't know which ones that we have coming in on the fruit, and it's very difficult for us to predict what they're going to do. But we don't want to kind of run them out of town right now, because we're like, but some of them are really good. Some of them give us beautiful aromas, some give us great textures. So what is it that we can do to optimize that biodiversity? And what that has allowed us to do is select the best of these non-Saccharomyces yeast and have them in a cultivated form so we can actually utilize them in winemaking. So we've got that predictability of the fruitiness, we've got the predictability of the antioxidation, we've got the predictability of all the good things that they can do without all the nastiness. So people like to say, but wine is a natural product. A microbiologist will always argue back and say a wine is not a natural product, a wine is a cultivated product. The natural endpoint of a grape-based fermentation is vinegar. So our job as winemakers is to produce wine, not vinegar. And if we take that a bit further, our job is to produce an aromatically well-balanced, texturally pleasing alcoholic beverage. And our non-sac kind of help us do some of that. So they help us enhance our wine quality by acting as antimicrobial agents, by acting as antioxidants, by being able to increase our aroma compounds, these compounds that are our primary aromas that originate in the grapes, like the terpenes and the thiols, but they can also produce fermentation-derived aroma compounds. They can enhance our mouthfeel. They can help to clarify the wine through their extracellular pectinase production. They can modulate the acid composition. And we all know in Texas, right? You're missing a little bit of that thing called acid. So our yeast can actually add back some acid into the matrix. And anytime we're diverting anything away from sugar, we could potentially lower the alcohol concentration. Let's kind of look at some examples of how we are using these new technologies, these innovations, in order to increase our wine quality. So the first organisms we'll look at, the non-conventional organisms, are from the Mechanicalia genus. Anytime in microbiology we have an SPP after the genus name, but we're not telling you the species, but because there's two P's, we've got multiple species. So in this instance, I'm specifically going to be chatting about Mechanicalia pulcherrima and Mechanicalia fructicola. So what we know with these organisms, they can act as antimicrobial agents. When we look at when we're talking about the non-sac that come in on the native flora and the fact they can produce a lot of acetic acid, about 30 to 50% could be Hanseníaspora clepora. Another 30% can be Candida piceus, and the other 20% is just a mix of everything else. And those Hanseníaspora cleperas, piceus candidas, do produce a lot of acetic acid. So just by adding in a cultured Mechanicalia into our matrix at a fairly low dose, 10 grams per hectoliter, we end up with an implantation rate, or if we look at the microbial population, 97% of the microbial population is our Mechanicalia, and only 3% is the Hanseníaspora. But when we don't add this organism into the matrix, then our Hanseníaspora is half of the population and other yeasts are half the population. So right there and then, we're using our organism as a biocontrol agent, the same as what you do in the vineyard with your IPMs. And how it's working is it's removing something from the other yeast and the Hanseníaspora that they require. And the thing that they require in fairly high levels is oxygen. Normally, we would use SO2 to control pre-fermentation microorganisms, pre-fermentation oxidation, but it's not that effective because of the level of sugar. So what we'll see is when we measure the dissolved oxygen level, our control in our orange line and our SO2 at 50 part per million kind of look the same, and you're no change in your DO initially, then it just kind of rises over the period of 5 days, but it kind of levels out on day 2 there up to saturation. Whereas with your Mechanicalia, the oxygen is utilized really, really quickly, leaving the environment O2 depleted. And the reason it scavenges that oxygen so quickly is to produce polyunsaturated fatty acids for its plasma membrane. So it's outcompeting the other organisms through oxygen utilization. What it can also do is scavenge the iron from the system, and in scavenging the iron, it produces a pulcherrimic pigment, pulcherrimic acid, which is antimicrobial. So it is antimicrobial against other non-Saccharomyces, but it has no action against Saccharomyces. So again, the Mechanicalia is becoming dominant through different means. Mechanicalia fructicola, similarly, when it goes into the system, it outcompetes the Hanseníasporas, the cleperas, the piceus, the candidas, as well as acetic acid bacteria. And we see a big difference in our post-fermentation volatile acidity. So The Mechanicalia species, the commercial names are Initia and Gaia. They both work by really quickly colonizing the juice mast and or removing essential nutrients and or by producing mycotoxins. So they're able to exert a fitness advantage or a dominance over other non-sac yeast and bacteria. So essentially they're becoming the top dog per se in the environment. So they work great as antimicrobial agents. They can also scavenge copper, thereby working as an antioxidant. We can use them as drivers of aromas and flavors. We can use them to reveal nor-isoprenoids and terpenes, to reveal thiols, or to produce esters. So your Mechanicolia pulcherrima has enzymatic activity that Saccharomyces just doesn't have. Saccharomyces is selected for its ability to ferment. That's your main selection criteria with Saccharomyces. But with our non-Saccharomyces, that's not our main selection criteria. We're looking for how they can enhance quality. So our terpene is bound to two sugar molecules. It's bound to generally a pentose as well as a hexose. And Saccharomyces doesn't have the best ability to reveal these compounds. So your terpenes are compounds that could be minty, floral, citrusy, spicy, even a little resiny. In the grape, they're mainly in the conjugated form. They're bound up to the sugar. And when they're in the conjugated or bound form, we can't smell them. They are sensory neutral. So the enzymatic activity of your Mechanicalia can produce the arabinofuranoside as well as the glucosidase. So it cleaves off those sugars. So it takes it from a non-adiferous compound to an adiferous compound. So something you can't smell to something that you can. So it increases the aroma capacity and potential of your terpenic varietals. But arabinofuranoscidases and glucosidases are not the only enzymes that Mechanicalia produces. It also produces a lot of carbon beta lyase. And it produces, if this little blue line here is how much Saccharomyces produces, this green line is how many units of enzymatic activity the Mechanicalia produces. You'll see that, I mean, that's a pretty linear production. It produces quite a lot of enzymes, and what it does is it cleaves off thiolic compounds in that you find in things like Sauvignon Blanc, Semillon, Pinot Gris, oh gosh, Rieslings, Grenache, Syrahs, Merlots. They're all conjugated, so you can't taste them when you walk the vineyard. If a winemaker walks the vineyard, tells you they're tasting their Sauvignon Blanc, it tastes like passion fruit. They are just telling tall tales. You cannot taste these compounds because they're bound up to amino acids. Well, the Mechanicalia has the ability to produce the enzyme that cleaves off the passion fruit or the citrus compound from its amino acid base, so it becomes aroma active. So again, enhancing varietal expression. Toidalspora delbrueckii produces ester. So if you look at how much ester is produced compared to control, you get, and it's not the fact that you have one ester gives you one aroma. This is just an indicator compound for aromatic expression. So we tend to see when we have higher amounts of certain esters, we pick up higher levels of certain flavors, right? But it's not a one-to-one ratio. So we'll see when we have a Troilospora in the system that we have higher apple pear notes, higher apple banana notes, not much difference in peach expression, higher pineapple pear and higher citrus red fruit. So we're just having an all over increase in our fruit expression. So again, we could get, by using our biodiversity that's naturally in the grapes, using our selected cultures, we increase aromas and flavors. We could also modulate mouthfeel. So when mouthfeel can be, we can build mouthfeel using a lot of different compounds. Polysaccharides is what we think of the most, or manoproteins, which are polysaccharide. But we also could have these polyols, these C5, C6 compounds. And we see when we look at these C5 and C6 compounds, when we have Saccharomyces alone or Tarlospora delbrueckii in conjunction with the same Saccharomyces, well, we don't have much difference in our xylitol, a little bit more sorbitol, a little bit more mannitol, a little bit more ribitol, but significantly more arabitol. Now, arabitol is helping to build volume and sweetness in the mouth. Okay, so we're impacting texture. We can use them to modulate our acids as well. So it's again different non-Saccharomyces, and within these, we can't take the same information for the genus and species we've been discussing and apply it to all of the organisms because there are definitely strain impacts. But what we'll see when we use Lachantia, or if you learn microbiology back when I did, this was known as Clebromyces. So Clebromyces thermotolerans, or renamed Lachantia thermotolerans, will take that glucose, that fermentable sugar from the grape, and instead of like Saccharomyces that puts it through the Ebden-Meyerhof pathway, ending in pyruvate, which will go to ethanol, it will do a direct glucose to lactic acid production. So for every gram per liter of glucose this yeast consumes, it will produce 1 gram per liter of lactic. So that's quite significant. So we'll see that when you're producing more acid, it can have an impact on your pH, so an acidifying action. How much it will impact your pH is dependent on a lot of other things, especially the buffering capacity. But in this Tempranillo, it lowered the pH from 3.5 to 3.4. We increased our titratable acidity expresses in tartaric from 6.3 to 9.7 because we got 3.3 grams per liter of glucose consumed. Now, if you don't control Lachantia, you can get up to 9 grams of lactic acid produced, which can be a little bit too much. Lactic acid gives a freshness to the wine. It does not give any dairy qualities, but you definitely want it to be in balance. You don't want to produce that much lactic acid that you throw off the mouthfeel of your wine. Another organism, this is the Schizosaccharomyces, which would be the US pronunciation, or Schizosaccharomyces pombe, is the non-Saccharomyces yeast that's been around the longest. This has been out for decades in an encapsulated form. Saccharomyces does not have the best ability to metabolize malic acid. The pathways for malic acid degradation are just not operational fully when Saccharomyces is fermenting. But Saccharomyces will use a little bit of malic acid and it will go malic to succinic acid. Schizosaccharomyces pombe can utilize quite a bit of malic, 2.3 grams per liter of malic, and it will give you about 0.1% ethanol. So it goes from malate through pyruvate through acetaldehyde and out to ethanol. And this is something that is used very frequently in cool climate winemaking to try to balance the malic acid. Because in cool climate winemaking, the malic acid can be in excess of 6 grams per liter. In hot climate winemaking, where we are, right, you guys and me, excuse me, we don't have a ton of malic acid, right? Our malic acid is actually quite easy to manage with Enococcus emi. But in some situations, the malic acid is so high, you cannot degrade it all through malolactic fermentation. So this is another route for demalication. So our non-sac yeast, we're just at the tip of the iceberg with them. They're really interesting because they can really change wine quality. They can improve wine quality by controlling the spoilage organisms, so giving you all the benefits of our non-sac, all the when we're able to exploit all this different enzymatic ability, all these different pathways, we're able to bring different things to our wine that we have not necessarily been able to bring. We can control oxidation, we can enhance varietal compounds, produce textural compounds as well. But there, excuse me, but there are huge strain impacts as well. Non-sac, as amazing as they sound, right? They're like, wow, super innovative, super cool. They could do all these things. They've got a couple of little flaws as well. They're not tolerant to alcohol, right? They cannot conduct the alcoholic fermentation. You still need your Saccharomyces to conduct the actual fermentation. The other thing is they're really difficult to get into an active dried form. They're very difficult to produce. So a lot of strains don't make it all the way through to the commercialization because they're either not genetically stable or they cannot be produced in an active dried form. So when we're looking at all these non-sac, right, we're able to actually have a much better prediction now of having more of the symphony rather than the spoiled grapes. But remember, they are only impactful in the first stage of fermentation. Once you're starting to get to 4% alcohol, they're all going to start to die off. Right, so 4% alcohol is about 9 bricks consumption. We've still got a lot of sugar left to consume, and that is what the job of Saccharomyces is. So we're talking about innovation. We're talking about all these cool organisms and what they can do. Well, we're not in a place that we can use one of these to replace our Saccharomyces. We cannot use these today to produce a non-alcoholic wine, right? They just don't have the capacity because the alcohol itself is toxic. So we're still using our Saccharomyces to consume sugar, and from sugar we'll get ethanol, glycerol, and CO2. How much glycerol you get depends on the strain, depends on the temperature, depends on the starting sugar, depends on your fermentation management. But we'll also get a whole host of metabolically interesting compounds, right? So just because we're talking about innovation and, biodiversity and exploration into new organisms, we've still got a lot to learn about Saccharomyces, which is our conventional organism. So it is our fermentative strain of choice. It still has a huge role to play in winemaking, but we've got more than 300 different strains that are currently available. And that goes to show the genetic diversity in those strains. If we kind of look at that dendrograph, you can kind of see how closely they're related versus how far away they're related and their interrelationships and where they come from. But there's a huge amount of diversity within the species. And if we utilize that diversity correctly, there's no end to knowing what they can do. People will often ask us, well, why can't we go in and change its alcohol production rate? Why can't we go in genetically and change its sugar to ethanol ratio? Why can't we take those 17 grams of sugar that normally gives us 1% ethanol and maybe have it consume a lot more sugar and give us a lot more alcohol? We can, absolutely. That is able to be done in the laboratory. The wine tastes terrible, right? So that's not our goal, right? We still want to produce something sensorially pleasing. So we're stuck with our alcohol at this point, but we can definitely reduce our alcohol using our non-sac. So our Saccharomyces, our main fermentation organism of choice. Remember, it can also reveal non-odoriferous varietal compounds due to its enzymatic activity. The enzymatic activity of Saccharomyces is not nearly as great as the enzymatic activity of non-Saccharomyces, but it can still do it, and we've been exploiting it. It can produce a lot of aromatics via the precursors that are present, right? If you've got a lot of precursors for esters present, you get a very, very fruity wine. If you don't have a lot of the precursors and you don't treat your yeast properly, you'll get a hot, bitter, alcoholic beverage. Okay, so you got to make sure your yeast is working in your favor. And to work in your favor, because the conditions must be correct, that we're not going to go into today. So like the non-sac that can reveal a lot of terpenes, your Saccharomyces can also reveal some of these monoterpenes. So remember, these are your floral compounds, the fruity compounds, the spicy compounds that are mainly available in that conjugated form, that non-odoriferous form. So some of your Saccharomyces has the ability to release the terpene into its odorative form. We think some Saccharomyces strains can actually produce monoterpenes through de novo synthesis, but there's a lot of work to understand those pathways, how exactly it produces some of these compounds. We really understand a lot about thiols. So those are the varietal compounds I was saying are in Sauvignon Blanc is the poster child for thiols, but it's available in Pinot Gris, Semillon, Chenin Blanc, Riesling, Syrah, Grenache, Pinot Noir, anything that you could think of that is either tropical citrusy or blackcurrant. You're going to get the thiol. So again, they're unexpressed in the grapes because they're present in their conjugated or bound form with either cysteine or glutathione. And your Saccharomyces strains with the beta-lyase activity can release them. We used to call this beta-glycosidase activity until we actually learned it was not, but these compounds were not bound to sugar, they were bound to sulfur compounds. Your yeast are all have this very similar capacity for thiolic uptake. The yeast will take up the thiols during the very early stages of fermentation because they're going after the amino acids, unless the conditions are so that the amino acids are not able to be taken up by the cell. So you've got too much ammonia in the system, for example. So all yeast, under the correct circumstances, will take up these thiols, but only if the yeast has that internal enzymatic ability can the thiols be released. And when we're talking about thiols, we're talking about the 4-MMP, or they're now sulfur compounds, so the slide's a little bit older, that would be the SSP. And that can be like a really fresh aroma to boxwood, to gooseberries, and the over 40 crowd call it cat pee. Okay, so never give away your age by describing a Sauvignon Blanc as having a cat pee aroma. Or it could be the 3MH, which is the passion fruit citrusy notes, or its acetate form, again, passion fruit citrusy notes. So to reveal these compounds, your Saccharomyces can do it, but you've got to treat them correctly. And what we'll see is there's a big strain variation in that expression. So all these can take them up, not all the yeast can release them. And this is some work that was done by Henty Schweiger back in 2006 in Australia. It was when they were trying to develop some yeast blends. And they were looking at 4-MMP, 3MH, and its acetate form, 3MHA. And that 4-MMP, as I said, is boxwood, passion fruit, and blackcurrant is how it shows itself in reds. The sensory threshold is 3 nanograms per liter. So you'll see that some strains are quite below. Some are a little above sensory threshold, and some are much higher than the sensory threshold. Likewise, for your passion fruit, grapefruit, or what we would say tropical citrus, sensory threshold is 60. So most of these strains were above sensory threshold. And the acetate form, so your 3MH being esterified, going to the ester form, again, the very low sensory threshold. And you'll see that most strains were above, other than L22, 26. So we're kind of looking at trying to understand how the yeast plays a role in thiolic expression. As a word, Vin7 has a little genetic quirk, and if you stress her out, she will produce a lot of acetic acid. So we generally don't even talk about Vin7, but we don't cut out data because it doesn't make our point, right? We show our data and we make sure we've got an explanation for it. But in order for you to pick up these thiolics, compounds from a sensory standpoint. Some new work out of OSU showing that the esters must be present. So you've got to have other esters that are generally fruity as impact aroma compounds to kind of lift the thiolic expression. So esters, Saccharomyces can produce esters. Different yeasts have different ability to produce esters. And it's if the Saccharomyces has the enzyme operating systems, the correct enzyme operating systems, which are controlled by the AFT1, the acetyl transferase 1 and 2 genes. So you've got to have the genetic material there, and then you've got to have all the correct precursors available. So you've got to have the nitrogen there. So your amino acids will go through the Ehrlich pathway. They'll end up as higher alcohols. They'll combine with acetyl-CoA. You have a little bit more transformation to get your acetate esters, and your ethyl esters will be generated through carbon metabolism as well. So our ester expression, again, definitely impacted by your yeast, but really what we're starting to learn, some newer work coming out of INRA, is showing that our ester expression is really impacted by the types of amino acids when we feed the yeast amino acids. Ester synthesis is a biphasic production. You'll have some esters produced while the yeast is growing, which is from the begin, like 2 to 3 Brix drop to 1/3 of the way through fermentation. The yeast stop growing at that part, but you've still got quite a lot of sugar left. And at that rate, the nitrogen is just being used for cell maintenance. So there's more nitrogen that could go into other pathways because initially all that nitrogen is going into growth. And then those other pathways that the nitrogen is getting punted into is ester production, or that's one of them. So when we give our yeast amino acids 1/3 of the way through fermentation, we get more esters. But we also see that we need biotin and thymine for ester production, and lipids, the natural lipids from the grape. If your lipids are too high, we suppress ester production. So we're looking to have about 2 milligrams per liter of the grape sterols for aroma production. And obviously you're going to control your environmental conditions. Different aromas will be produced at different temperatures. So that exploration in Saccharomyces, using our heat maps, we can really see all the different yeast strains and their relatedness and their response to different conditions, whether it's environmental stress and nutrient stress, but I put it in because we can kind of get an idea of all the different aromas that the different strains can produce under the different conditions. So I like heat maps because they're a really quick and easy visual until you start to dig into the data, then they give me migraines. But you could really see if each line is showing you, kind of giving you an indication of the yeast and a genetic background, so how it's responding to the different environments. So with that, oh my goodness, 3 colleagues have called me. With that, hopefully I was able to extend some of the excitement about this different biodiversity, different innovation, different biotechnological exploration. With you now, those of you who didn't know, the non-Saccharomyces yeast, as well as bacteria that we did not talk about, are associated with the grapes. Some of them could be really interesting and really enhance wine quality. Some could be very detrimental to wine quality. But we're at a place now where we are actually selecting, cultivating, and adding non-Saccharomyces yeast based on their special abilities. Because Saccharomyces is not actually associated with the vineyard. It's not something that we will find when we take our grape samples. It is an organism that is associated with the built environment. So in our instance, it's the winery, but it could also be the brewery, the cidery, the bakery, the distillery. Just not something that is ubiquitous. It has a very, very narrow ecological niche. It is a contaminant, if we want, right? It is something that's not native to the grape, but it's the contaminant that we really get to exploit. And if you've got your right conditions, you've got your correct yeast there, they do have the ability to enhance your wine quality, whether it's through helping to enhance the varietal compounds, to produce aroma compounds, or to impact mouthfeel, whether it's through building volume and palate texture, or acidity and freshness. So selected cultures make you sure that you've got the organism there you want. So you're taking away the unpredictability, you're taking away the chance. Some winemakers like to think of the beginning parts of winemaking as expressing microbial terroir. Well, that's great when you've got estate fruit, you've got very controlled temperatures, you've got very, very controlled environments. But when you're picking and transporting and not able to ferment straight away, that microbial terroir could end up as vinegar. So we're actually working to protect our grape quality. So with that, thank you for your attention. I'm going to pop out a share. Things might freeze again for a minute. And I'll be able to see all your lovely faces and we can chat for a little bit if you'd like.